you guys in the audience see me? I'm kind of sitting behind the camera, all right? And I'm trying to avoid the light. So this meeting is being live streamed. Wonderful. Oh, there we go. Yeah, there we go. All right, thanks, Jonathan. Cool. So thanks everyone for coming out. It's really nice to see you. It's nice to be back to in-person events. We're excited about that. As Beth said, my name is Julie and I'm the CSO at IndieBio. We have an office in New York, we have an office in San Francisco, and both of them are working with companies at the most cutting edge of technology that can be commercialized. And I'm really excited to be here, in part because I've been a part of the Genspace community for a while. Genspace is where I first did Gibson assembly. It's where I first performed CRISPR. I've been in and out of the community for a long time, so it makes me very happy to be able to bring some of these cutting-edge technologies to the broader community, where hopefully you're going to hear something tonight that will make you say, wow, I didn't know that was possible. We have a trio of CEOs who will be speaking about their journeys, about their technology. We're really going to focus on the science and how it works, and what you can build with fundamentals of biology. Let me first quickly introduce IndieBio. Easy fix. IndieBio works with the world's best scientists to turn them into entrepreneurs. 70% of the companies, the startups that we work with, are helmed by people who are coming directly out of their undergrad, graduate, postdoc, and in some cases even professors who are taking the research that they've been working on and putting it into the commercial front. And so we work on folks in terms of business development, IP strategy. We make sure that they meet certain milestones during our 4-month program that sets them up for continued growth after they leave. Yes, picture today. I have a picture up here. I have one of the CEOs who will be speaking. How do we do that? We work with people throughout 4 months, but I would say even before and afterwards. We form long-term relationships with all of the startups that we work with. During the program, the companies will receive $525,000. There's a lot of mentorship, and I'm saying that from my end, and you can ask the CEOs what that relationship is like. We have rent-free lab space in the Upper East Side, and we're super excited for our new space, which will be at 710 Plaza at 31st Street and 7th Avenue, starting next year in 2022. So that will be very exciting. And if you have any interest in biotech and forming a startup, I mean, I have the application website up here, but the most important thing is to talk to the IndieBio team. We're happy to even kind of position folks and say, look at
Who's that? Who's that? The CEO.
Today, we are going to be speaking with 3 amazing CEOs. We have Danielle Rose, Uyen Tran, and Harvey Tran, who are going to talk about ag tech, material science, and genomics. I'm looking at Harvey in the back, right? How do we frame this? Genomics. And I'm very excited to ask them some questions. The way that I've framed this evening is mostly me asking them questions. We want this to be interactive. I mean, we have a live event on purpose. So if you have questions, we have Layla on the chat, and she will be monitoring if you have any particular questions. We'll ask a couple of questions from the audience afterwards, after each CEO, just to make sure that we, answer the things that you came here to find out in addition to the questions. Come on down. Yeah. Right. Oh, it's really great. But it's good. It's much more visible. All right. So the CEOs actually did not prepare their own decks. Usually everyone prepares a deck and has this very refined pitch. But what I did instead was to come up with a couple of images that I thought would be interesting talking points for each of our speakers today. So this should be fun for both of us, I hope. I have no idea what to expect. Yes. One thing that I know about Danielle is her passion for commercializing. And so I have a picture that might be a little bit triggering here. And I would like you to tell me what it is about this picture that you find so frustrating.
So the long and short of this picture would be that in biology, there's this fun thing that you learn about in microbiology classes called the Great Plate Count Anomaly. So that's kind of this fundamental understanding that exists in the field that we can only culture about 1% of microbes in the lab currently that exist on our planet. And that's very disproportionately skewed to certain environments. So like we're really good at culturing human gut microbes, but if you go to say the soil or the aquatic ecosystems, that kind of is a disaster. And people have been kind of complaining about this problem. And it's like, wow, like we really don't have access to like all of these microbes. And I learned about that in my undergrad. I'm like, wow, like that means that all of the technology we have is coming from a very small subset of microbes that we have access to. So like, what do the other 99% have? So the main thing that kind of is accounting for this Great Plate Count anomaly, in my opinion, is the fact that we still do this. We still culture microbes the exact same way that we have for 150 years, and which is basically on Jell-O made with soup broth, and we're surprised when things that like boil over. Which at like a fundamental understanding level kind of makes sense that it would work. It's like trying to take polar bear and put it in the savannah and being surprised.
So what is it about this field that you think is the biggest problem that has in the field that you're in?
Yeah, so definitely two things. So one is a fundamental shift in how we think about microbes. So thinking about them more like we think about animals and less like we think about, say, like this thing that's microscopic that we don't really consider as a living thing almost in how we treat it. So I think that definitely a brain shift has to happen in trying to learn techniques. As well, like, dear God, we need more automation.
Automation, like for example?
Oh yeah, like for example, plates are a great example. If you're doing polyethylene for like plates, it's you in a hood, hopefully not an anaerobic hood, because like then it's even worse. Like there for hours going like pick microbe, streak on plate, pick microbe, streak on plate. And the entire time you're thinking it's like, wow, everything I've been doing, a robot could do better and faster. And I just spent 8 months doing this. Let alone people who spend their entire PhDs doing that. So like a human shouldn't be involved in that process. A human should be using their brain to think about how to like create new technologies and use the microbes to change the world. They shouldn't be sitting for like 8 hours a day in the lab.
All right, wonderful. We're going to put a pin in this because we are definitely going to revisit automation. Can you introduce Ceragen, the company that you're starting, and the problem that you're solving?
Yeah, so at Ceragen, we develop probiotics for plants. So these are microbial inoculants that are made of beneficial symbiotic microbes that are plant growth promoters that we use to basically help farmers reestablish the natural, healthy symbiotic communities of microbes to improve their crop yields and thereby producing more food.
One of the analogies that you use, that I've heard you say, is that plants and their microbes are very similar to bees and plants in terms of how they evolve together. Can you talk about that?
Yeah, so there's definitely a lot of symbiosis that happens with these kind of communities. And so these microbes have evolved with these plants. And so now you have certain things like different pathways and like genetic pathways that are kind of in two organisms instead of one, the same way that we need bee pollination. For example, the ethylene stress pathway in plants is a really good example of that. So here it is.
We didn't plan this.
Yeah, so
So the folks online are probably having a hard time hearing this?
Okay. So ethylene stress is a kind of generic stress response in plants. So when the plants experience that stress, say a heat wave, then there's a little bit of a chemical called ACC that is in plants that gets converted into a little bit of ethylene. And that's what you see in that first little beneficial spike.
I've heard about ethylene. That's a good thing, right? It helps plants to ripen.
It is a plant growth hormone. It is a plant hormone involved in a variety of different genetic pathways, and one of them being the stress pathway.
Okay.
So in this instance, Little bit of ethylene, good, creates beneficial stress response, plant's happy. But then plants do this thing where they create like a whole bunch more ACC as part of this process, and then they convert all of that to ethylene and cause this massive secondary spike, and that can be days or weeks after the first stress event. And that causes leaf yellowing and leaf loss and like growth inhibition and like in extreme cases death. So basically the plant self-destructs. So, and partly that's because if these natural plant growth promoting microbes aren't there, that's what it does. If these microbes are there, they sense that increase in ACC, they start producing an enzyme called ACC deaminase, breaks that down, and they use it as an nitrogen source, and then you don't get that secondary spike. So it's kind of a regulatory pathway that evolved in two organisms that like, if one's not there, which is what happens in some of these agricultural scenarios, like hydroponics, which is what we work in, where these plants have very reduced microbiomes.
I really just picked up a thing. I looked for tomatoes in greenhouses. Okay, misleading photo, but yes, this leads us to hydroponics, where there's no soil microbiome.
Yeah.
So how do plants interact with microbes in that kind of environment?
Yeah, so plants in hydroponic systems have very minimal microbiomes. So you can kind of think of them, we experience the same issue in lab mice as well. So laboratory mice have very reduced microbiomes, and it actually is a whole thing that causes all sorts of issues with the reproducibility of animal tracks. So that's a whole separate problem. But in plant trials, basically, or yeah, in these hydroponic grow systems, a lot of soil microbes can't survive in hydroponics. Kind of just think about it, it's really not the same environment at all. Very waterlogged, soilless substrate where you're getting all of your nutrients through a nutrient solution. So basically liquid fertilizer. So it's not really surprising that a lot of microbes can't make that transition. So what we do is we find the microbes that can, that do still have these beneficial symbiotic relationships with plants. And then we basically create these probiotic solutions that farmers can add to the plants over the roots and reestablish these colonies.
And tell us about your first product and why how it interacts with plants on a long-term basis where other probiotics.
Yeah, so especially in the hydroponic space, since a lot of these microbes can't survive very well, you have to continually replant, so they just die. Unlike that, our product, since it is selected for one that proliferates in hydroponics, it's a single application at the beginning of growing season. And for something like greenhouse tomatoes, which you can see here grow its very large vine, it's not really what you think of in your garden tomato. Those growing seasons are like 8 months. So it's one application at the beginning of the 8-month period and not again until the next growth season. So that's super handy for the farmers.
So how are you going to know if this? How do you know if this stress is less of this ethylene or ACC deaminase being made by microbes?
Yeah. So the way that the farmer tells is more fruit. The plants will be healthier in a stress event. We won't get as much leaf yellowing. For tomatoes, under stress conditions, tomatoes will get something called blossom end rot, which basically is a very literal description of it rots in the blossom end of the tomato. So making it not sellable and not useful for consumption. So helping reduce all of those symptoms is kind of what the farmer would see on their end.
How are you testing other farmers right now?
Yes, we are. We actually have 3 commercial pilots ongoing. So 2 of them are smaller pilots that we started over in January, and then 1 is a larger 2-acre pilot that we started in August.
Excellent. So that's for tomatoes specifically?
Yes, that one's specifically for tomatoes. These products do need to be designed for use in each plant. Microbial products and microbial symbiosis in general is very specific to the type of plant it works in. So if you had like a garden and you planted a tomato and a cucumber and a lettuce plant, and then you sequence the microbiomes, they would be very distinct depending on which root system those microbes grow.
That brings us full circle to the automation question. So how are you going to find new microbes for the lettuce, for green peppers, for eggplants? now that you have some for tomatoes, can you use those?
Yeah, so we can't use the tomato microbes that we have, but that doesn't matter because our company has basically developed a agnostic pipeline that we can use to develop microbial products for any type of plant. So we use a combination of novel culturing techniques and high throughput automated screening, as well as whole genome sequencing to basically screen these plants, screen these microbes, figure out which ones have the best capabilities for being plant growth promoters, and then using those for plant trials and developing products.
I heard the first thing that you said there was plating. So you are not completely able to get plating out of the system.
Yeah, so as far as like, we do use some plates, you can see like sometimes I'm working on it. That's my dream, to get rid of that and never have to see it again. I just haven't quite got there yet.
To not see any plates or to not have humans making the bacterial aggregate?
I mean, if you can get rid of them entirely, I think that would be better. But yeah, at least no human involved.
Has anyone in the audience ever used a bacterial plate, like to either do colony counts or get some kind of
Yeah. You never like had to select for novel qualities. Like if you're to pull something off of like in my undergrad, I spent 8 months doing that with like various. I had 16 different types of media doing culturing for the gut microbiome under aerobic and anaerobic conditions. So it was hundreds of plates that I would pick up, and it would look kind of like that, except less colorful. And I would be like, which of these are different? I see 8 different varieties of white blood. So like, it's really not something we should do.
what is changing in terms of the automation side and looking at some of the other behaviors or characteristics of these different groups?
Yeah, so whole genome sequencing is definitely something that's very novel in as far as who the company is. So we use long read sequencing, in-house sequencing, which I think is like something that you couldn't do 5 years ago. Like it took a while for the price point to come down and for the accuracy of those sequencing systems to really catch up to a point that was viable in-house. And like I think that's going to be something that's really
Bigger picture questions about what or Ceragen as you.
So as far as like our dream, when I go back to kind of what I was thinking, I was sitting in university listening to about all of these microbes, like I just remember thinking to myself, I'm like, this is so cool. We can harness these microbes to solve so many problems. So my real dream for Ceragen is just to solve as many problems as I can by harnessing these really cool microbes. So getting it onto as many plants as possible and affecting as much of the food production chain as we can.
So challenging? Is that or is it just, oh God, I can't think of a single thing?
Oh no, startups are accurately described by most people as the hardest thing they ever did, and it just keeps getting harder. And I would say that that's pretty much true, unless you've gone through some pretty big hardships in your life prior to that. So like I'd say everything's hard, but the hardest part is just keeping going. Because like when you're in a startup, it's a very unique situation that's definitely not for most people. So it does take a particular breed of crazy people to this, where like there's no one else. You wake up in the morning and you're like, okay, like these 10 things need to get done, and you look at the list and you're like, well, I only know how to do half of these, but they all have to get done, so I guess that's my problem. So you have to teach yourself what you don't know, and you're 100% accountable for everything. So like if you need to order marketing materials, guess what? You're the marketing person that day. Something goes terribly wrong in the lab. There's no PI to fix it for you. Like that's your job.
Yeah. Right. What is the motivation for you to, waking up every day, doing 10 things?
Yeah. Part of it, I love the challenge. I'm just one of those people that thrives under that kind of environment where I can try new things and learn new stuff. And like that to me is just, it's solving that problem is really rewarding. The other thing is like ever since I was little, I've always like wanted to make a difference. I've always wanted to help change the world. And I think startups are a great way that you can actually do that. As like one person starting a movement and really have a movement.
In terms of the impact that you're having, what do you think? Okay, so let's say all of us go to the grocery store and are like picking up, plants and different produce. How is it affected by what Ceragen has done?
Yeah, so you won't know. You'll never be able to tell what happens on the back end. So that's actually something that's true with a lot of synthetic biology or any sort of biological innovation. A lot of time the end consumer would have no idea what was happening in the supply chain. But what we would see is those like fruit, they might be slightly better quality, and the price point might get changed a little bit, but overall there will just be more availability. We won't have to use as much land to produce it. The farmers will be able to make more money when they're doing so, and so the economics of the supply chain will be.
Does anyone in the audience are online? I can see that there's like comments that I can't read them. So Leila, if you can give voice to the online.
Questions about anything?
Yeah.
A little bit special sauce. The long and short of it is we do a little bit of everything. So yeah, as far as like what we're trying to develop, it's just really trying to harness a lot of these novel tools and things that are kind of that we now have access to that we hadn't before, or can bring into the space from other industries like engineering. Like my co-founder is an engineer. And partly the reason for that is there's technologies that have existed as commonplace in the engineering world for decades that, like, after growing up in a family where my dad's an engineer, my brother's in engineering, seeing the tools they had access to, and I was like looking at what we had access to in the lab, and it's like, this is absurd. Like, why are we doing this? Like, this is 20 years behind where it should be. And I remember having conversations with people about like automation. It's like simple as something, and someone like just getting into weird conversations where it was like someone's like, yeah, we can't automate this because how would a robot be able to tell if a plate got set down wrong? And it's like pressure sensors. That's how that's a thing that exists. And it's like the disconnect there, like I find like crazy. So I think there's like huge room for disruption and having this intersection of like multiple different disciplines to basically work together to really create some major innovation.
Other questions? Are there any from online?
Yeah. Are you using MinION sequencing?
Yes,
Yeah.
love MinION.
Yeah. Okay. Good.
Yeah, I remember learning about them in university, and when we like started our own company, I was just so excited when that arrived. So that was kind of like the little scientist nerd dream come true.
Danielle, it's a pleasure to have you. This is for the last one. I'm sorry. This is what do you dream of building? Apologies. Right. When can you talk about Tômtex? Yes, okay. Can we talk first about the name Tômtex? And what is the origin of its name?
So Tômtex actually like not Tômtex, but actually like Tômtex. So text, everyone knows like textile, like Tôm with the simple facto is English. So I'm Vietnamese and I was born and raised in Vietnam, and this is the original of the name.
my boyfriend is also Vietnamese. He always tells me I'm saying his name wrong when I say Tômtex.
Tômtex.
Tômtex, yes. All right, so can you tell us about your experience in Vietnam before you founded this company, seeing some of the waste produced through industries that we here in New York City might not see in our everyday lives?
Yeah, it's a long story. So I was born in and raised in Da Nang, Vietnam, which is like a beautiful coastal line surrounded by the beach. And so basically, I deeply impacted by the textile flooded into my hometown, as well as like I live quite just 2 to 3 blocks out of close to the leather manufacturing. And it's quite devastating to see my hometown contaminated by the act of leather tanning and textile pollution. So when I was young, I don't have any new clothes to wear. I wear all of the second-hand clothes. Oh, don't get me wrong, I love all my clothes. I cherish all of them. But so I learned. I'm a fashion designer, by the way, and I learned actually like all of the brand, like Ralph Lauren, Gap, all the famous brand by using second-hand clothes. And that is when I dreamed to become a fashion designer and come to America. Yeah.
You mentioned in there that there was a leather tanning was a source of pollution. Can you talk a little bit about what that is? So we start with like animal skin and then what happens afterwards?
Yeah, so actually like leather is the most harmful environmental textile to produce. Turning skin to leather use a lot of dangerous chemicals like tanning agent and chromium. And when you do that and then you flush it back into the river and it's contaminated the whole region of the city. And at that time we don't have like high technology just to reduce the chemicals. We just flush it and discard it into the natural environment.
That sounds really not ideal. That sounds like a bad system. On this slide, I also have a picture of shrimp, which you said is the origin of the name Tômtex, right?
Yeah.
So what is it about this particular organism, this shrimp that we see here, that is part of that story of waste?
Yeah, so actually, in fact, that 50 to 60% of stream is actually shrimp, right? And a lot of culture doesn't consume this byproduct. And particularly, the food industry generates like 6 million to 8 million tons of shell-seafood waste every year. And just in Vietnam,
what would that look like 6 to 8 million tons? Is that like a football field? Do you have any idea about how much that is and how many?
yeah, football field is the great sample.
Yeah.
Like maybe couple tall buildings combined together every year, and it's a tremendous, like source for our ingredients. But specifically, like in Vietnam, we have around like 400 to 500 kilotons of stream byproducts every year, and this is the waste. And we see it as an opportunity to get the parmesan out of places as our raw materials.
So can you introduce Tômtex now? Tômtex, and talk about how you are using some of that material to create your, explain what your company is doing.
Yeah, so we are designing sustainable materials for the future, and we start with leather alternative first. So our material is 100% biodegradable, and this can be an alternative to faux leather and animal leather made by seasonal waste and mushroom.
Excellent. We're going to get into some of the process of that, but you did not immediately form Tômtex from Vietnam. You took a kind of circuitous route. You mentioned you're a fashion designer.
Yeah.
So how can you tell us a little bit about your road from Vietnam to becoming a fashion designer?
Yeah, so basically, like, I'm in love with fashion because, like, I learned all of the brands by the secondhand clothes. And when I, like, come here, and my dream is to become a fashion designer. And actually, I work in the fashion with some of the luxury brands in New York. And I'm like, I know where's the problem come from. And our, like fashion industry system is not, cohesive together, it's not connecting together. And the way we do it is like we're promoting the linear kind of behavior as consumers. Like we buy one time, we wear it one time, and we just discard it. And so where we discard it, all of the unwanted garment and unwanted, unsold clothes, we be either incinerated or discarded to the landfill, or flushed back to Vietnam or Bangladesh or Africa, South Asia, and hit like military cycle. And that is the switch point that I want to somehow, like maybe create some sustainable materials for this brand to use, so they can create more sustainable products.
And I remember in conversation with you and some other people previously, we were talking about wouldn't the large brands themselves push this sustainability forward? Someone else said this, not Gwen. And then Gwen was there in the background like, no, they wouldn't, because we were working with some really well-known designers. I could not find that dress, so we'll just pretend this is the actual dress. But there was a dress that you designed for a very well-known designer.
Yeah.
Can you tell us about the material that was sourced, that you wanted to be sourced, versus what they ended up using?
So yeah, so basically, like as a fashion designer, like 3 years ago, we have very difficult time to sort out like sustainable or natural fiber yarn material in general. It's always like in the back rack with like very expensive price point, not fitting our budget. So as a designer, I have like very struggling finding sort out the sustainable materials for my design. And yeah, a really, a last draw for me is when I designed a runway collection with the designer, Alison Van, and I basically want to design a dress choosing the silk materials. And because like we tie them and convert it, and we want to drive that profit more, get more profit out of the dress. So that the dress that I designed is actually about $70, and we still using viscose for the materials instead of silk. And at that point, I was like, okay, sure, like we agreed to do that. But in the end of the day, the dress selling like the best seller across the world, and we produce around 10,000 to 15,000 back dress long. So I was like, if this, dress doesn't make out of natural material, it's nice gonna end up. Like people gonna buy it, it's gonna end somewhere, it's not gonna be great. It's really another problem that we have to tackle in the future.
So the high-end design use viscose. What happens to viscose when it's done?
Yeah, so basically like viscose is like kind of synthetic fiber, and viscose doesn't biodegrade in the life cycle of this product. So, like we either like again like burn it to regenerate energy, which is not safe to burn anyway, or it just lay there and it's not gonna biodegrade.
The skills to become a material designer, what do you do?
Yeah, so that is the last job for me. I'm like, okay, I don't want to do this anymore. Because a lot of people doesn't want to tap into the textile design and doing fabric coordinating. So that's when I enrolled the new school of design and learning about natural materials in general. And I fall in love with like polymer and learn more about it, and then just like do a lot of experiments in the lab, as well as like learn other things like natural material as well, and come up with this material. So that is the foundation of Tômtex.
Let's talk about how you make your material from chitin. So chitin is in the shrimp shells, right?
Yes.
Okay. So we've got a lot of different steps here. Can you walk us through how you go shrimp shells to, I believe this is actually a picture of your material up here, right? This is your real material?
Yeah, it's my favorite material.
Yes. So it's very flexible. Obviously, shrimp shell is very hard. So what do you do?
Yeah, so basically, like we are turning, seafood waste like stream shells, lobster shell, crab shell, like tiny, we can obtain it from those shells waste as well as mushroom. So we're using mushroom. No, we're using phytosan. So phytosan is the second most abundant biopolymer, and we're using phytosan to mix with other green substance or biopolymer through a strong cross-linking process to produce our materials. And the process, the formula is very versatile. So we can, later on, maybe we can produce like yarn fiber with this same technology.
So here, but you haven't actually, these are not your, this is not your bio yarn.
No.
No, but this is your biomaterial.
Yeah.
Yes, okay. And, so chitosan, the chitosan, how is that related to the?
Yeah, so phytosan actually like it's just a part of the string. Like string shell is you can like obtain other products out of string shell as well. So yeah.
So can you tell us about the material, the first materials that you're making?
Yeah, it's actually pretty interesting because I have kind of I'm separated from science. I come from design background, so everything surrounding me, like I want to make like to turn it into something that functional or that more like, so when I produce my material, basically it doesn't have any transparency. It's like plastic-like material. So when I feel it, I think it's like very strong, flexible, durable, and I put like let experiment some colors and like some texture within the material to see like if like behave and if the outcome is what I'm looking for. And I learned so much from the material. It teach me like the versatility when I put in colors, when I put in different texture design that I design. It just adapt everything that I design. So that's why I think these materials can be so much potential in the future. That it be a lot like give the designer as well as the scientist a lot of room and creativity to
Very excited. I would like to, we forgot to bring some with us. So we've seen some of these beautiful materials in the lab and office. Let's take a step back and talk about your dream for Tômtex and for building a big company. So what do you dream?
yeah. So within Tômtex, I envision the world where people surrounding themselves with high performance materials sourced directly from waste and not fossil fuel.
And you have very strong opinions on waste. You want to share your opinions on waste?
So I actually I don't believe anything at waste because I just like we learn a lot from nature and we take advice from nature. And in natural world, there's no such thing as waste. So for me, that I don't believe in anything like designing anything that lasts forever. I want to design something that is come back to the earth, bacteria can grow in it, animal can fit on it, and I see the word as ecosystem that that like everything that have the function can be a part of that. So I don't believe in.
It's a strong belief. It's a good one. So same questions I gave to Danielle. What is the most challenging part of building a new startup?
Everything. So I think like startup in general, the experiments, no one teach us at school. Basically, this is the whole new skill. That's when like I feel very lucky to get into IndieBio and get the guidance and even small step like how I can talk to lawyers, how I can talk to investors, how I can better communicate with the customer and creating customer problem. So as a founder, we wear many hats and we need to, yeah, like Danielle said, wake up every day, have a list of everything, and just keep doing it. There's one way, there's no way back. And the company is like depend on you basically. So I feel very, responsible what I do, but at the same time, very exciting.
What excites you and keeps you
Yeah, I think the more exciting thing for me is like this sustainable movement that it's not like a trend anymore, it's like what people need and how it's survival like sustainable shift is, like in addition to is good or is the right thing to do for the planet, but also is the right thing to do for the business. So people like designer, business, scientists working together across country to bring this together is, I think, it's very exciting to.
Thank you, Gwen. Questions from the audience? So that's a couple questions. So can I think that you actually mentioned this, so can your material be transparent?
Yes, it's at first it's like transparent. It's like bioplastic kind of thin like material. So there's no colors, there's no textures, and yeah, it's transparent. They form. Yes. What does that mean?
It's like something like a glue, something sticky that is like.
Oh yeah, I know it's hard to say in Vietnamese. I don't know how to say in English. Actually, it's not like a glue. Like the material actually a liquid form that basically you mix with water, and when water evaporates, you have the material left. So that is what I would process like. Yeah, so good question. So basically, like this can form. I have the mold, and then you put a liquid onto it, and then it can form whatever it can capture all the texture detail within that. Yeah, very good questions. So we are at a very early stage. That's why like we have to start somewhere first, right? So that's why like I reached into like Vietnam first because like we have tight connection with the supplier. So the supplier that we working with is they collect all of the seasonal waste from the manufacturer, and then they take it to back their um, facilities, and then they create a design. But yeah, our big dreams is to have the model that we can apply in different regions. So we in like conversation with people in like Netherlands or North or even in America, for example, to somehow like build up pipeline and use each different waste stream to produce the material. But it's a complex system. And like there's not one size fits all, and we need to investigate each different maybe city or country to do that and collaborate with the local business or the local waste management system. So, but potentially, like if we can figure it out in Vietnam first, we have the great foundation to take it and fly to different countries.
I get one more question. So, and you are distribute more, focus more on the bacteria itself and enable the production for different companies to use it, or rather just focusing on the collection of products made for this material.
I would, I love to be just a supplier. Yeah, I want to make this material accessible for everyone, and I'm not gonna turn it into brand of products. We really producing the material in general, and we like more material, sustainable material. But yeah, I would love to like working with all the brands across all the industry, the fashion, interior design, industry, and the material can be run into different economic products. Sorry, we need to dismiss additional questions. Here today is Harvey Tran, Inso Biosciences.
How are you doing?
Good.
You enjoy the other conversation?
Yes. Now I'm prepared.
Good. We're ready to talk about microbiome.
Yes.
Right. We're going to jump right in. You're going to be so happy. I've pulled a lot of things on your slides. Can you talk about, first let's start with the 3 different aspects of genomic sequencing. So first let's define what genomic sequencing is. We're going
Sure.
to talk a lot about that.
Yeah. So we talk a lot about genomic analysis, but when people say genomic analysis, it's really broken down into 3 parts. First, you have to process and prepare the samples, get the DNA out. That's the sample prep section. Then you have to sequence the sample, read that DNA, get the information. And then you have to make something with that information, get analysis, analyze the data, get the information from whatever information is.
Can you, let's say, in 2 or 3 sentences, the history of advancements in these different
Yeah. So I think sequencing is probably the one that you guys are hearing about in the news. So I think back in 1990s, about 30 years ago, people started what's called the Human Genome Project. They were going to sequence the entire human genome. And this is an international project that took 10 years and almost $1 billion to accomplish. Sequence the human genome for the first time. And it was all over the news. It was a big deal. But then you fast forward to today, you can sequence the human genome in just a few hours. A single scientist can do that. It costs less than $1,000 to accomplish. And people are pushing that down towards the $100 genome. So $100, everything about your genetics. So it's very powerful. The costs are dropping extremely quickly for sequencing. The accuracy is extremely high. It's already pretty much at 99.98% or something like that. But on the data analysis side, yeah, that covers the sequencing. On the data analysis side, the number of pipelines, the number of tools that have been developed over the last 10 years for analyzing sequencing data have been growing exponentially. So that side is also growing extremely quickly. But then you look at the sort of the last piece of the sample prep, and you look around at what people are doing when you talk to them. They're using methods that are 30, 40, 50 years old, and it hasn't kept up. And that's kind of what we want to address as a company, is the sample prep side.
Awesome. So obviously, the bottom half of this slide
Yes.
addresses what it is that people are doing. Can you talk, I mean, just briefly, what, as many of us, raise your hand if you've done some sort of sample prep. You've gotten a piece of DNA out of some cell. Yeah, not only
Great.
for raising hands, but for those of
Yes.
you who are in the digital world, we're getting thumbs down from the audience as well. Why are you getting thumbs down from the audience?
Yeah, well, I'm actually glad to hear that everybody's somewhat familiar with this process. So I think the overall way that I would summarize this is that the way that we're preparing samples is just not in a scalable system, right? It's not a scalable format. Like, it's okay to do one or a few samples like that, but as sequencing is improving so quickly and data analysis is improving so quickly, what's happening is people are wanting to do more and more sequencing, which is reasonable. And people want to do these projects with thousands and thousands of samples. And in order to accomplish these projects, you would have to prep samples for months. So that's not something that is going to scale into the future. And if you think about genomics right now, it's starting to get out there more because of everything that's happened around COVID, but it's not really out there yet. It hasn't been sort of rolled out yet, right? And if you look at the computer field, the way that I think about it is like computers, they couldn't have made the impact that they have right now if they weren't made accessible. If you had to be a computer scientist and it took you like 4 hours to figure out how to like write down open a program or something like that, it wouldn't work, right? So I think that's the same thing for genomics. It's so many steps. There's so much labor involved, and you have to be so trained in order to know how to prepare samples. And even people who are trained, who do this every single day, have a lot of trouble creating that consistent, high-quality sample. That's what we want to address.
Sounds to me like the answer is automating. Why don't we just use the pipetting robot? What are the other problems that are inherent with preparing DNA from cells?
So automation is something that I think people are reasonably thinking about, and they have built these, very complex automation systems, essentially $500,000 to $1 or $2 million large laboratory tools kind of sitting there. It's about the size of that table. And they can automate parts of this process. They can take out the DNA and purify it. But in order to go into some formats of sequencing, you have to process the sample additionally. You have to shear the DNA to the right sizes, you have to select the right size, and put that in the sequencer. Those are things that automation today can't handle. And automation itself, when people are saying that, they're really thinking about robotics, which is taking the same hand processes and adding robots and replacing those. But there's no new technology. So everything that is wrong with this technology that's 30, 40, 50 years old is still carried over and compounded by the robotics. Robots aren't as precise as humans sometimes. Yeah, if you think about it, like it's somebody who has like a decade of experience preparing samples, and they know for each sample how much to tilt that test tube in order to draw out every last drop of the thing that they need to purify out, versus a robot that's just going to do it the same way regardless of how things turn out.
As someone who has done
Maybe as
hundreds of
AI.
these, all right.
Maybe as AI improves,
Yeah.
yeah.
All right, I like it. Okay. All right, I think we've laid the groundwork for what the problem is. What are you building at Inso Bio that will help to address some of this?
Yes. So we are building an automation system, a single system that will cover the entire range of what is sample prep right now, handling all the way from taking a sample in to pulling out the DNA contents, preparing that DNA, purifying it, sizing it correctly, pulling it off the system, and for that. So we're basically taking 7 to 11 steps of labor, about 4 to 6 hours of total time, and bringing that down to one step and 30 to 40 minutes total time, so about a tenth of the time. And we're taking that hands-on time part where people are, literally at the bench processing these things. Right now, it's about 1 hour per sample. So we're going to take that down to about 5 minutes.
But, I don't know if this will actually play or no, I think, there we go.
Oh, great.
Okay. So can you explain what
Yes.
is happening in this video, please?
Yes, so on the video on the left, basically what is happening are human cervical cancer cells being loaded onto our technology. So what we have built is called a microfluidic technology, and it utilizes tiny, tiny channels of fluid in order to handle biological materials in an extremely precise way. So the unique component of our technology is what you see these little dots here. That's what we call a micropillar array. It's like a forest. You can think about it like a forest. And the pillars are smaller than the cell. So we load the cell in, and then once the cell is loaded into the pillars, we can pop the cells. And the popped cells, they'll essentially disintegrate and disappear, but the DNA contents of the cell will become entangled in this micropillar array. That's how we extract the DNA. There's several very sort of technically unique functions of this. Like we can purify the DNA in solution. That goes to our name, Inso. We keep the DNA in solution. So that's very unique. That helps the DNA quality. We're able to extract the DNA physically, so there's no in-channel chemistries that's required to bind the DNA, catch it. It's just purely physical process. So it's very fast. It's very efficient. The way I like to describe it, as you guys can see that spaghetti on the floor. That's there to just give the concept that it's because the spaghetti is so long and it gets caught on the fork. There's no binding to the fork, right? And that's the same concept of what's happening.
Really serendipitous origin story that I don't think you often use.
Oh yeah, I don't tell
Can
that.
you just tell us how it was you even discovered this technology worked?
Sure. Yes, I was a first-year student, a first-year graduate student at Cornell, and I had joined Harold's lab, my PhD advisor for this work came out of, and we were tasked with building these microfluidic channels and using a way to catch cells and to separate that out from the DNA that we're going to pull out of those cells. So we didn't have this entire pillar, right? We only had like a few rows of it. And we would catch the cells and we would see it go in, and then we would put in the chemicals to pop the cells, lyse it, and we would find no DNA on the outside. And we're like, what is going on? Like, we just can't find this DNA. And we would try to repeat these experiments, load in more cells, but still nothing came out. So it was just really like perplexing to us. By the time we loaded this kind of cells, we should have seen something. And that's kind of when we're like, well, I think we just got to take a look and dive the DNA, find out where it is, just track this thing the whole way through. And that's when we found out all the DNA was caught on these pillars. We're like, oh, we found it.
So you really just wanted to build a cell lysing machine.
Yeah. Actually, we weren't set out to like try to do all this. Like we kind of stumbled upon it, and we're like, well, what can we do with something like this?
What can you do with something like
What can you do with something?
this? Hey, I've got this DNA. Now what?
Yes. Well, so it's part of our sort of multi-process system that really combines all of those steps of sample prep, condenses it down to just one system automating the entire thing. So now that the DNA is gotten to this step and it's entangled in the microbiome, right? What we've been working on as a company, as a startup in the last 2 years, has been chopping up that DNA to the right sizes so that it can fall off, essentially, of this pillar array, and then we can collect it for sequencing. So an example of something that we've been able to accomplish is taking a single finger prick droplet of blood, right? Squeezed, and this is a co-founder's finger droplet of blood, loaded directly onto our device with no pre-processing, with no centrifugation, none of that work that typically has to happen in the hospital. No pre-processing, one droplet of blood loaded onto our chip that's like this big. And from that, we can process the sample in about half hour and sequence the entire human genome from that one droplet. That's an example of something that we can accomplish. And we validated this through triple-fit experiments, showing 99.6% of the DNA mapped back to the human genome. So there is a lot of data that we have. And we're thinking about what is a market that we can go after first. And I think that's a lot of the navigation that we've had to do as a company is we can't necessarily go after these big hospital systems first because there's a lot of FDA approvals, there's a lot of regulatory hurdles. Selling into hospital systems is extremely difficult. We need large sales teams, long sales cycles. We don't really have all those resources. So I think for us, it was a real kind of journey of figuring out what is the application that is the low-hanging fruit, that we still provide an extreme amount of value in, that we could convince customers to be torn away from what their comfort zone is to try out a new technology. And so for us, I think we're actually converging was talking about the bin ions and all that stuff. So we've shown that, from our system to go all the major systems, those are all systems that.
Instead of, let's say, a Ceragen kit, which I'm looking at directly, like a shelf full of Ceragen kits and all the different prep kits.
Yeah.
Do you expect, like, in the future, a place like Genspace would just have like all of these? I don't know what's it going to look like. Is it going to be a tube? Is it going to be a box? Is it gonna
Yeah,
be
ideally it would be something like I don't know, like a desktop PC sized thing, and it would process all your samples for you. I think it's analogous to like a thermal cycler for PCR. Most laboratory systems have one, and it's not like in front. It's just kind of in the background, but everybody knows how to use it. Everybody knows how to interact with it. It's there, and you know that if you need to do PCR, you can go to the thermal cycler. That's been standard for decades. So I think that's kind of what
You actually answered my next question. Do you expect to buy this?
Yeah.
Which is, I mean, everyone eventually, but obviously
Yes.
not at first. So, what is it you're hoping to build with Inso Biosciences? What is the big picture?
Yeah, that's a good question. I think for us, we definitely, obviously we would like to be this ubiquitous piece of instrument that's in genomic laboratories, that is sort of the go-to standard way of processing samples. That's a given. But I think if I were to sort of venture a little bit further, and this is maybe beyond kind of Inso as the company, I think what the future of genetics is going to be, and where this technology will be by the time it is sort of at the impact scale of what computers are now. I don't think that there's going to be one separate technology to prepare samples, one to sequence it, and then some other thing to analyze it. I think it's going to be like a giant player like a Roche or a Thermo will come in and vertically integrate the whole thing into one single system. It's going to be this black box, and samples are going to go in, and nothing will come back out except for the data that you are requesting from it. So I think everything will be in a singular place. There won't be any separate processing or something like that.
Same question I gave to Danielle and to Uyen. Most challenging part of building something there.
Yeah, well,
Yeah.
I think for me, it's probably. I think for me, it's been like managing emotions has been kind of a thing to figure out, because I feel okay. So I'll just give an example. 3 weeks ago, we were working with a grant writer to submit a $400,000 grant and then back-to-back submit a $1 million grant. And we contacted them for about like 2 weeks, and they didn't get back to us at all. So for 2 weeks, and this is like a month ahead of when the grant is due, and we haven't started writing it. And so for 2 weeks, we're just like, why aren't they responding to us? And you start feeling really down. Like, oh, damn, there's another one that's gonna ignore us. And you just like, because I think you're just like so in the process of building this thing every day that you don't have that time to like necessarily self-reflect a whole lot. And I think I need to work on that. Maybe I need to like meditate more or something. Yeah, you just like get caught in the emotion of what's happening like in that moment, and then like. At the turn of the dime, like they get back to us and they're like, "Oh, we've actually been working on it. We've made a ton of progress, and things are on track." And it's all like our co-founders and I were like, "Oh, we better scrap this. We better think about a different way to get funding." Like we're already creating like these contingency plans of what we're going to do. We're not going to submit this thing. So just having everything flip and us just being like suddenly in a good mood. It's just like, and then the next day you're in a bad mood, and the next day you're in a good mood. I think that thing has been like just. An interesting part of the experience.
Yeah, crazy that someone would not get back to you for 2 weeks in this world.
Yeah.
that's the crazy part of that story.
Yeah. I think on mine maybe at some different times.
Most rewarding part of building something.
Yeah. I think it's like the challenge, the people you meet along the way, the unpredictability. I think I like waking up to that.
Yeah?
Yeah.
Okay.
I mean, I know I just said that. I'm emotional. I don't know. I guess some part of me just likes that.
Yeah, I can see that.
Yeah.
Excellent. Thank you so much for answering these questions. Are there questions from the audience? From online? Yeah.
Yeah.
It's like I can sit down and computer and like write so I'm wondering, so do you think that when you were saying you want ideally your product to be bought by everyone, do you think everyone can have? Or do you think that very good is like I could go inside of my next kitchen and like see what's like, just for to look at it or something?
I think that's a great question because what you're touching on is something beyond just the technology. I think there's when you start thinking about like human health and genetics and all these things, you really are starting to get into this gray area of like A, is it regulated? B, is it like people want to know that information? Like I think there's a lot of people out there who wouldn't want to know their own genetic information. They don't want that kind of predetermined thing in their mind, and I'm one of them. So I think that's a good question. I think I think I would answer that and say we'd like to build something where if that's the world we live in, then yes, we'd like to be on kitchen countertops as well. But if it's not the world we live in, then it should be the ubiquitous instrument and where it belongs.
Oh, Harvey, you said something in conversation with me, I've heard you say, about what the future of genomics looks like and what tools kind of thing. Can you talk about that a little bit? Because I think that might maybe address this future world that you just kind of alluded to. Like, no washing your hands here. You got to actually pick a side. Like, is this something that people should have access to in our kitchens or wherever? Or is it something that you're hoping will remain in this medical kind of like laboratory sense? And we're talking like 20 years, so
Yeah.
Who knows? It's all kind of just speculation,
Okay, yeah, I guess that just makes sense. It's like, I think like if we were in a room back in talking about what we think computers should be, that people will ever need a portable computer. I think that's kind of like what the infancy of this entire thing is right now. There's no way I think that we could predict by building computers that would be TikTok or Instagram or all those things, but it enables things, right? When you build the tools and you put it into people's hands, they can go and build the things that you can't even imagine right now. And I think we're very much in that stage. When we can lay the foundation of how to build computers and how to build the internet and get the infrastructure down, then other people can come after us and really change the world.
I love the TikTok of genomics.
Yeah.
I want to put it that round. Are there questions from online, Leila? Yeah. Yeah, so
Okay. It's a very good question. I think for us, the primary weakness of the technology is having everything in a solution, right? Like, things need to be in a kind of aqueous format in order to be loaded into the microchip. So you can start with different types of samples, like tissues, or plant tissue, or soil, to get that tissue or soil or whatever it is to some kind of solubilized format first before it can be loaded on the chip. But if you can, and there's more and more protocols now, if you can, really confident that and going back to like blood, for example, the way that we're able to handle just a drop of blood so efficiently is because the way that the microfilters are set up, they're spaced so that it only captures certain sized things. White blood cells can capture the thousands and thousands of red blood cells flow right through. And that goes for plants as well. We can capture just the plant nuclei if we really think nuclei. But if there's a ton of starch, which oftentimes you have in plant samples, the starch will fly. So there's a lot of unique technology capabilities. Thank you.
I'd like to thank all of our CEOs for coming and having these conversations. So we do another round of applause. And I don't know if you want to say anything in closing. I will say this is one of the first hybrid events I think that Genspace has had. So I hope you guys online have been able to. I'm talking to my computer. I think it should be talking to this thing. Yeah, the mechanisms might not be 100%, but hope everyone online enjoyed it. So nice to speak.